Hello, welcome, and thank you for having us here. We're here from the University of Oxford to discuss how we're using Nextflow pipelines in our lab. And I'm here today with my colleague Patrick.
Good morning, everyone. So my name is Patrick. I have a medical background. I've come to do a PhD and acquiring a lot of skills in immunology and increasingly bioinformatics.
I'm Eleanor. I'm a postdoc within the Knight Group. Currently, I work on COVID. Mostly to do with vaccine immune genetics, but I have a background mostly in veterinary-based research, and I have to admit I am probably one of those scared of the black box scientists that we've all been discussing recently. So today we're just going to give you a brief talk. We're going to focus on the kind of interests that we have within the Knight Group, why we moved to Nextflow and how we're trying to implement those pipelines into our lab, and then a little bit about how we're using germline, somatic variant discovery and vaccination datasets, a little bit how we're understanding epigenetic features in a sepsis dataset, and then we're going to summarize this all up together. So the Centre for Human Genetics is where we're based, linked with the University of Oxford, and we belong to the Knight Lab. We're a relatively small lab as teams go. Anytime we have between 20 and 30 members. This ranges from professors to postdocs to undergrad and PhD students, all with a background that's quite mixed. We have some clinicians, some non-clinicians, and predominantly we're all wet lab-based scientists. Our main interests really are looking to understand how genetic variation impacts appropriate immune responses to a whole range of diseases. These include COVID, sepsis, ankylosing spondylitis, among many others. And predominantly we're using large next-generation data sets to do this, and these range from anywhere from 10 participants to about 5,000. And at the moment we're using a high-performance computer cluster with a Slurm scheduler, and historically we've had a lot of bash scripts that haven't been necessarily well maintained. They have a limited version control and they don't have much dependency management.
So I've been very interested in bringing Nextflow into the lab as part of my PhD. I think really for the reasons that Ellie mentioned. So we have had, at times, excellent bioinformaticians, but then gaps as well. And we inherited a lot of scripts without much in the way of modern software engineering practices, and we don't really have the resources to implement them ourselves. So Nextflow, and in particular the NF Core community pipelines, are very attractive to us. They really offer the opportunity to standardize the pipelines across different projects within the lab, often where we're doing the same relatively kind of straightforward things on large cohorts, and it's very important to maximize reproducibility as much as possible. We've also found, with many students in particular coming into the lab from wet lab and clinical backgrounds, it often can be a pretty steep learning curve, as everyone else has mentioned, getting to grips with the command line. And using Nextflow, and particularly the NF Core pipelines, is really right in the sweet spot where we can bring someone in and within days or weeks, they can be seeing great results from their data sets.
Okay, so briefly a little bit of background about what we work on. I'm not going to spend much time talking about COVID-19. I'm sure we all know it. I'm sure we all felt the impacts. But it is important to consider that vaccination was a huge effort to control the pandemic. And as of mid-November, about 13.5 billion vaccine doses were given across the globe. A massive effort by research scientists across the world in order to work out how we make these vaccines, how we give them, what they work for and who is protected. But actually when we consider this, there's still a huge number of people across the world who don't mount a good immune response to COVID vaccine. You either have underlying health conditions, age is an impact, and most importantly, human genetics underlie reasons why people have a low immune response. So one of the main questions that I'm working on in the Knight Group is there a genetic association to lower response to vaccination? And in this instance, it's related to the COVID vaccine, but really more broadly, do we see any reasons in multiple modalities why that might be the case? So we formed the COVID-19 Vaccine Immune Genetics Project we call COVID, in collaboration with the ONS, which is the Office for National Statistics in the UK. We invited 5,000 participants who either had a low or high antibody response. So when the pandemic kicked off in the UK, the ONS made a long-term study where they enrolled participants from about 500,000 households, and every month you had an antibody test. So we're using data from that to try and split our cohort into really low or non-responder and really high max responder. And we've collected quite a lot of data. So we have genotyping data, HLA typing, whole exome, and DNA methylation. And for this project, I'm hoping to use these Nextflow pipelines to really understand this whole exome and this DNA methylation. So quickly, we're just using the sarek pipeline here to answer questions like, are there rare and limited variants found that cause functional changes in proteins relating to antibody binding that could impact vaccine response? Do we see these variants in just the low responders And if so, could we classify these? Could we understand that bit better? And also, are these low response phenotypes driven by really rare penetrant genetic variants? And do these contribute to this increase or decrease in antibody response either way? So at the moment, I've been running the NF-core sarek germline pipeline to analyze these whole exomes. I have about 750 whole exomes, and I found that sarek and Nextflow is great. You can put them all in, it puts it all together, it has all these nice modules. I've personally been using multiple haplotype callers, so I'm using the deep variant, the freebase, and the haplotype, and I'm concatenating these together. And then it's great that you can have that with VAP already plugged in, as well as the associated plugins that I can then add on to VEP. And at the moment, I'm looking into whether I could also use the NF-Core's rare disease pipeline. I found this is a really great way for someone who's predominantly wet lab like me to sort of get into this field, to understand better, and with such a great community to learn from.
Thank you, Ellie. So a little bit of background on my project as well. So I'm interested more on the epigenetic side, looking at the causes of immune dysfunction in the period after critical infection. So this comes out of really the clinical and epidemiological observation that people who survive critical infections show changes in their immune function which can persist for several months after they leave the ICU. And these are measurable, but the next step is really understanding the mechanism behind them. And we aim to link these phenotypes to measurable epigenetic features, so particularly DNA methylation, histone modifications in different immune cell types, which culminates on the question of what are the epigenetic consequences of sepsis. And the resource for this is our sepsis immunomics project. So we're getting to around 400 participants, and we have then performed a number of multi-omic assays, so particularly DNA methylation, transcriptomics, and somatic variant-oriented target capture sequencing. And really for each of those blocks, I've found that the NF-core pipelines have just been fantastic for accelerating the analysis that I need to do. To give a quick sense of what this looks like, so this boils down to the question of which DNA methylation sites vary between our post-sepsis patients and then age and sex-matched controls who we've also recruited and sampled from. I found that at every step of the way, the methylseq pipeline has supported what I need to do. So we're using an assay which is relatively new, so a targeted enzymatic methylation conversion assay, and all of the features for a target assay are really there, and I've been quite surprised by sort of how little I've had to add on so far. We've also found that it works just perfectly with our existing cluster. So we've scaled up from dozens to now hundreds of samples without any drop-off in performance. And we see that the data that comes out of the pipeline is of excellent quality, so very nice methylation QC, and we're starting to see some very interesting epigenetic findings which we'll follow up with further functional wet lab experiments. So in conclusion, we think this has probably been a fairly light talk, but just to give a sense of what Nextflow and in particular the NF-core pipelines look like when they're deployed in a kind of medium scale, mostly clinical, partly wet lab group like ours. It's been a fantastic tool for improving reproducibility and scalability across our analyses, and has really allowed us as predominantly wet lab biologists and clinicians to go a lot further with our analyses. And it's been great to be at the conference learning a bit more from the software engineering side about future directions.
We have a lot of people to thank contributing to all our work here, but I would also like to thank Pat. Pat brought Nextflow into the lab. He's helped me train on Nextflow. I think it's been a really great addition to our team, and it's been really nice to be here and listen to all the talks.
Thank you so much, both of you. Very interesting talk. Do you see any, I may have missed this, but do you see any opportunity to contribute back some of the pipelines to NF-core or something like that? Would you be interested?
Yeah, certainly. I think that's something we're looking to do more of as we gain experience and then can start to suggest extra things. But to be honest, we found that almost everything we need to do is implemented really well already. So
that's wonderful.
Yeah.
Thank you so much, both of you.
Thanks.
All right.